Imagine a world teeming with life. So small, it's just completely invisible to the naked eye, yet capable of profoundly impacting our health. We're talking about fascinating single-celled organisms, protozoa, and today, specifically, flagellates and ciliates. Today, we're diving deep into their intricate forms, their life cycles, and really how they interact with human health. For this deep dive, we've gathered some great material that will guide us through this. Think of it as your shortcut, maybe, to being genuinely well-informed about these often overlooked inhabitants. They're in our internal and external environments.
Exactly. Our mission today is really to pull out the most important nuggets of knowledge from these sources. We want to use some surprising facts and hopefully a much clearer understanding of what these tiny creatures actually are and why they matter so profoundly. You'll discover not just what they are, but why their unique biology makes them such significant players in public health.
Yeah, absolutely. So whether you're just curious, maybe you're prepping for a meeting where, public health or an environmental microbiology might pop up, this deep dive is tailored for you. The goal is to gain knowledge quickly, thoroughly, but without feeling overwhelmed. It's a journey into the unseen, really, where every detail reveals a piece of a much larger puzzle. Okay, let's start with the absolute basics then. What exactly are flagellates?
Right. So at their core, these are protozoa, single-celled organisms, and they're characterized by having one or more of these whip-like structures. They're called flagella.
Like little tails.
Exactly, like tiny biological propellers, essentially. They enable their mobility through, various bodily fluids or water environments. Some even have what's called an undulating membrane.
Okay, what's that?
Think of it like a wavy fin sort of attached along the body that adds to their propulsion system. It gives them a different kind of movement.
Interesting. And their general body plan, is it complex?
Well, for a single cell, it's quite defined. They usually have a distinct body shape, enclosed by a flexible outer layer, sometimes called a periplast. Often there's an anterior depression, a sort of mouth called a cytostome, for taking in food. Inside, you'll find one or maybe two nuclei, along with other specialized bits, things like blepharoplasts, axostyles, parabasal bodies. These are all internal structures that help them function.
So it's not just a simple blob. There's intricate machinery inside.
Precisely. And understanding this unique cellular machinery helps us understand how they thrive in very specific, often quite challenging environments inside us, often totally unseen.
Okay. How do they reproduce and crucially survive outside a host?
Well, what's truly fascinating is their survival strategy. They might look simple, but they primarily reproduce asexually. Often it's through something called binary fission, basically one cell splitting neatly into two.
So they can multiply quickly.
Very quickly. But here's where it gets particularly interesting. This is really crucial for understanding their impact. Many of them can form cysts.
Ah, cysts. I've heard about those.
Right. This process, called encystment, isn't just a basic function. It's a critical survival mechanism. It lets the parasite essentially shut down its metabolism, form a protective wall, and withstand incredibly harsh external conditions.
Like what kind of conditions?
Oh, think extreme temperatures, drying out, even chemical disinfectants that would kill the active form. This resistance is absolutely vital for ensuring the contamination of new hosts and the widespread of disease.
So without the cyst stage, they'd be much easier to control.
Dramatically easier. The cyst is the key to their persistence in the environment and transmission. There are only a few exceptions, like Trichomonas, which we'll get to, that don't form cysts.
Okay, so where do these flagellates fit into the bigger picture of life, classification-wise?
Sure. They belong to the phylum Protista. Specifically, they're often placed within the Phlasmastigophora, basically animal-like flagellates. And within that class, you have various orders. Important ones include Diplomonidida, that's where you find Giardia.
The one we hear about a lot.
Exactly. And Enteromas. Then there's Trichomonidida, which includes Trichomonas itself, and also Dientamoeba, and Retortimonidida, which has Chlamastix and Retortimonas.
So quite a few different types infect humans.
Oh yes, quite numerous.
Yeah.
And they have really varying levels of pathogenicity. Some, like Giardia intestinalis and Trichomonas vaginalis, are definitely significant pathogens causing real health issues. Others, though, are considered to have little to no pathogenicity. They're more like benign residents or commensals.
So it depends on the specific flagellate.
Absolutely. And it also depends on where they set up shop, whether they colonize, say, the digestive tract or the urogenital tract. That dictates the clinical manifestations you see. They have incredibly diverse strategies and impacts.
Okay, let's do our first deep dive then. Let's talk about Giardia intestinalis, also known as lamblia or Giardia duodenalis. You said this is a true pathogen.
Definitely a true pathogen. It's responsible for Giardiasis, sometimes called lambliasis. It's a really widespread intestinal infection found pretty much all over the globe. Affects humans, affects many animal species too.
And its impact, you mentioned it's significant.
Hugely significant, particularly for children and for people who are immunocompromised. In these vulnerable groups, Giardia can cause really severe digestive problems. It often leads to something called malabsorption syndrome. Basically, the body just struggles to absorb essential nutrients from food.
That sounds serious, especially for kids.
It is. For children, this is especially concerning because it can seriously affect their growth and overall development.
Right.
In some cases, it can even lead to stunting.
Wow. So it's not just a temporary stomach bug. It can have long-term consequences.
Precisely. It's not just a stomach ache. It can fundamentally alter a child's developmental trajectory if it's chronic or severe.
That's a critical point. What about its discovery? Is there a story there?
Actually, yes, there's a fascinating history here. If we connect this to the bigger picture of scientific discovery, the very first microscopic observation of this parasite, it dates all the way back to 1681.
1681. That's early.
Incredibly early, by Anton van Leeuwenhoek himself. And the really surprising part, he observed it in his own stool samples.
No way. Talk about dedication to science.
Right. But it wasn't until 1859 that a more precise description was provided by a scientist named Lamble. That gave it a clearer identity.
And you mentioned it's also called Giardia duodenalis.
Yes, and Giardia duodenalis is now understood to be what we call a complex of species. It's not just one single entity.
Meaning there are different types?
Exactly. There are at least 7 distinct genetic types, or assemblages as they're called, labeled A through G. Now, what's crucial for human health is that only genotypes A and B are commonly isolated in humans.
Okay, A and B in humans.
But here's the kicker. These same 2 genotypes, A and B, are also found in a whole wide array of animals. Domestic pets, farm animals, wild mammals.
So it can jump between animals and humans.
It seems so. This highlights its broad host range and makes control and prevention really quite challenging. It's not just a human-to-human issue. The other assemblages, C through G, tend to be much more host-specific. Like genotype E is pretty much only found in livestock like cattle and pigs. Got it. Okay, let's talk about its forms. You mentioned cysts earlier. Does Giardia have different forms?
Yes, it has 2 key forms, and each plays a vital role in its life cycle and impact. First, there's the active feeding stage called the trophozoite.
Trophozoite, what does that look like?
It's typically described as pear-shaped or piriform. Rounded at the front, kind of pointed at the back, measures about 16 to 18 micrometers long, so still tiny. It has this characteristic kidney-shaped depression on its underside, which acts like a suction cup to attach to the intestinal lining. It has 2 nuclei, which is quite distinctive.
And flagella.
Yes, it has a central rod-like structure called an axostyle and 4 pairs of flagella. These give it incredible mobility. When you see it moving in a fresh sample under the microscope, it often looks like a falling leaf tumbling through the fluid. Some even say it looks like an owl's head because of the 2 nuclei appearing like eyes.
Falling leaf or owl's head. That's memorable. And where do you find this form?
These trophozoites are quite fragile, actually. They don't survive long outside the host. You typically find them in the diarrheic, watery stools of infected individuals.
Okay, so that's the active form. What about the cyst?
Right. Then we have the cyst form. This one is more oval-shaped, a bit smaller, about 10 to 12 micrometers long. And unlike the trophozoite, it's immobile. It doesn't have free flagella.
What's inside the cyst?
Inside, it contains anywhere from 2 to 4 nuclei, depending on its maturity, along with remnants of the flagellar structures. But the most important feature is its wall. It's surrounded by this characteristic, thick, double-contoured wall. You can often see it clearly under the microscope.
And this is the resistance stage you mentioned.
Exactly. Here's where it becomes a major public health game changer. These cysts are incredibly resistant in the external environment. They are the form responsible for spreading the infection, for the dissemination and infestation of new hosts.
How resistant are we talking?
Critically, they resist most common disinfection methods. And this includes the standard chlorination levels used in drinking water treatment.
Wow, chlorine doesn't kill them.
Not reliably at standard doses. This isn't just some quirky biological fact. It fundamentally challenges our conventional thinking about water purification. It means that seemingly safe, treated water can still potentially be a vector for Giardia. It highlights a critical vulnerability in public health.
So what does kill them?
Only more extreme measures are consistently effective. Things like freezing temperatures or boiling the water.
That's crucial information. Okay, Let's trace the life cycle. How does someone actually get infected?
So the life cycle helps us understand the spread. Infestation begins when a person or an animal ingests these mature infective cysts,
How do they ingest them?
typically through contaminated drinking water or maybe food that hasn't been washed properly and has cysts on it. This is why it's often called a protozoan infection linked to the fecal-oral route, basically. Fecal contamination and unclean hands. Poor hygiene is a major factor.
Okay, so the cyst gets swallowed. What happens next?
Once ingested, the acidic environment of the stomach and then the conditions in the small intestine trigger the cyst to exist, basically. It opens up. It releases an intermediate form, which then quickly divides, usually resulting in 4 active trophozoites from each cyst.
4 from one cyst.
Typically, yes. These trophozoites then rapidly colonize the upper part of the small intestine, the duodenum mainly. They use those ventral suckers to attach firmly to the mucosal lining.
And then they multiply.
Yes, they multiply rapidly by binary fission, just splitting in 2, increasing their numbers. Then, when conditions perhaps become less favorable for them, maybe due to changes in the gut environment or the host's immune response starting to kick in, they undergo intense biological changes to encyst again. They form those resistant cysts, which are then passed out in the feces, ready to survive in the environment and potentially infect a new host, continuing the cycle.
It's a very efficient cycle for the parasite.
Extremely efficient.
So transmission is mainly oral, through water or food. Are there specific risk factors?
Yes, the primary route is definitely oral ingestion of cysts. Contaminated water is a huge one, especially waterborne transmission, which often leads to quite large epidemics in certain parts of the world. Poorly washed food is another common vehicle.
And reservoirs, where does it hang out?
The reservoirs are quite broad. Humans are a major reservoir, of course, but also various animals, cats, dogs, guinea pigs, livestock, beavers, are famously associated with it in North America. And naturally, contaminated water itself acts as a reservoir.
And who is most susceptible?
Several factors increase susceptibility. Young age is a big one, which is why children are so often affected and can get quite sick. Any kind of underlying intestinal problem can make infection more likely or severe. Severe malnutrition weakens the body's defenses. And certainly, a weakened immune system, whether from illness or medication, can lead to more severe and prolonged disease.
Okay, let's talk clinical manifestations. What does Giardiasis actually look like in a person?
Right. The symptoms typically appear maybe 2 to 3 weeks after someone ingests the cysts. But it's important to note that some individuals, especially healthy adults, can be asymptomatic carriers. They have the infection but show no symptoms, yet they can still shed cysts and spread it.
So they can spread it without knowing they have it.
Exactly, which makes control difficult. When symptoms do occur, it's essentially a parasitic duodenitis inflammation of the duodenum, and it usually presents in 1 or 2 main forms.
Okay, what are the forms?
There's the acute form. This often starts suddenly with things like upper abdominal pain, often crampy, significant bloating and gas, or flatulence. And the really characteristic symptom is diarrhea.
What kind of diarrhea?
It's often described as muco-greasy diarrhea. The stools are typically pasty, yellowish, sometimes described as resembling cow dung, unfortunately, and they often have a really quite nauseating foul odor. Importantly, there's usually no blood or pus in the stools, which helps distinguish it from things like bacterial dysentery or amoebiasis.
Got it. No blood or pus. And the other form?
The other is the chronic form. This is more persistent, obviously. It might involve frequent bowel movements, maybe more than 10 a day sometimes, often alternating with periods where the person has normal bowel movements or is even constipated.
So it waxes and wanes?
Exactly. The stools in the chronic phase remain generally soft, greasy, and foul-smelling. Patients also commonly experience ongoing abdominal pain, often worse after eating, along with weight loss and a loss of appetite.
And you mentioned the particular impact on children earlier.
Yes, for children, a really significant complication, especially with chronic infection, is that malabsorption syndrome we talked about. It severely affects the absorption of carbohydrates, fats, proteins, and even essential vitamins like A, B12, and folic acid. This malabsorption is what often leads to retard staturopondérale, basically, stunting or failure to thrive. It seriously impacts their growth curve and overall development. It's a critical long-term consequence for kids.
That's really concerning. How is it diagnosed then, if someone has these symptoms?
Diagnosis relies primarily on finding the parasite in stool samples. So it's a parasitological examination of fresh stools. Lab technicians are looking for either the mobile trophozoid stage or those characteristic oval cysts.
How do they look for them?
The standard method is direct microscopic examination. They take a small amount of fresh stool, maybe mix it with saline or iodine, and look at it under the microscope. This allows them to see the falling leaf movement of the trophozoites if they're present, or spot the distinctive cysts.
What if the parasite numbers are low?
Good point. Sometimes, especially in chronic cases or if someone isn't having active diarrhea, the number of parasites shed can be intermittent or low. So they often use concentration or enrichment techniques. These methods concentrate any parasites present in a larger stool sample into a smaller volume, making them much easier to detect under the microscope.
Are there other tests, like culturing it?
While direct examination, sometimes with special stains like MIF or Giemsa to help visualize the forms better, is the mainstay, culturing Giardia is actually extremely delicate and difficult. It requires specialized media and conditions, so it's typically reserved for research labs, not routine clinical diagnosis. Sometimes specific antigen tests or PCR tests on stool are used too, which can be very sensitive.
Okay. So diagnosis is mainly stool examination.
Yeah.
What about treatment and prevention?
Right. For treatment, a group of medications called nitroimidazoles are generally the drugs of choice. Metronosols, often known by the brand name Flagellate, is very commonly used.
How is it given?
It's usually taken orally for about 5 to 10 days, depending on the specific drug and the patient's situation. Dosages are adjusted for adults versus children. Sometimes, especially if symptoms persist or to ensure clearance, doctors might advise doing 2 successive courses of treatment, maybe spaced about 10 to 20 days apart. And ideally, a follow-up stool test should be done after treatment to confirm the parasite has been eradicated.
Are there alternatives if metronidazole doesn't work or can't be used?
Yes, there are other effective options. Tinidazole and secnidazole are related drugs that often have the advantage of being given as a shorter course, sometimes even just a single dose, which can help with people sticking to the treatment. Ortidazole is another one. For cases that are resistant to these standard treatments, which can happen, another drug called quinacrine might be considered, though it can have more side effects.
What if cysts are still found after treatment?
That's an important point. If cysts persist after a full course of treatment, it could mean a few things. It might be recontamination from the environment, maybe the person didn't adhere perfectly to the medication schedule, or in some cases it could indicate actual therapeutic failure where the parasite is resistant to the drug. That would require further investigation and potentially changing the medication.
Okay, that covers treatment. What about prevention, prophylaxis?
Prevention involves measures at both the individual and collective level. Individually, it really comes down to good hygiene. Thorough hand washing is absolutely crucial, especially after using the toilet, after changing diapers, and before preparing or eating food.
And washing food.
Yes, diligently washing fruits and vegetables, especially those that might be eaten raw, could have come into contact with contaminated soil or water. If you're unsure about water safety, especially when traveling or camping, boiling water for at least 1 minute is the surest way to kill Giardia cysts. Filtering water with a filter certified for cyst removal can also work.
And on a community level.
On a collective level, prevention involves ensuring safe drinking water supplies. This means regular and effective treatment of municipal water sources, using methods known to inactivate or remove cysts like filtration and sometimes UV light or ozone, as chlorine alone isn't always enough. Rigorous control of the water distribution system to prevent contamination after treatment is also key. And in some situations, like during outbreaks or in high-risk settings, systematic screening and treatment with asymptomatic carriers might be considered to reduce the overall parasite load in the community and prevent further spread.
Right, tackling it from multiple angles. Okay, that's a really thorough look at Giardia. Now let's pivot, as you said, to a completely different track of human health. We're moving from the digestive system now to the urogenital tract. Here we encounter Trichomoniasis, caused by Trichomonas vaginalis. You mentioned this is a highly contagious protozoan infection and a very common sexually transmitted infection, an STI.
That's right. And this parasite presents a really unique challenge compared to Giardia.
How so?
Well, what's truly fascinating here, and it's a fundamental difference, is that Trichomonas vaginalis is strictly human. It only infects us. And crucially, it exists only in its vegetative active form, the trophozoate. It never forms a resistant cyst.
Never. So no protective shell like Giardia.
None at all. And this immediately raises an important question, doesn't it? How on earth does it survive and transmit effectively if it doesn't have a durable cyst form that can withstand the outside world?
Yeah, how does it manage that?
The answer lies in its extreme fragility outside its very specific home, its biotope, which are the urogenital mucous membranes, the vagina, urethra, etc.
How fragile are we talking?
Extremely. It can only survive maybe 1 or 2 hours on a damp surface like a wet towel. It might last up to 24 hours in fluids like urine or semen if kept moist, but basically exposure to drying or air kills it very quickly. Its optimal growth conditions are also quite specific. It likes body temperature around 35–37 degrees Celsius, a slightly acidic pH, typically 5.5 to 6, and it prefers anaerobic conditions environments without much oxygen.
So that fragility directly impacts how it spreads.
Absolutely. It makes direct, intimate contact pretty much essential for transmission.
Okay, let's look at its morphology. What does this trophozoite look like?
The Trichomonas vaginalis trophozoite typically has a more rounded or oval shape compared to the pear shape of Giardia. It's roughly similar in size, maybe 15 to 20 micrometers long, sometimes a bit larger. And it's highly mobile. It achieves this movement thanks to having 5 flagella. There are 4 anterior ones sort of sticking out the front that are free, and then there's a 5th flagellum that runs backwards along the body, forming an undulating membrane that extends about 2/3 the length of the cell.
An undulating membrane again, like some other flagellate.
Exactly. This combination of free flagella and the undulating membrane gives it a very characteristic, often jerky or twitching type of movement when you observe it under the microscope in a fresh sample.
Okay. So, given its fragility, how does contamination occur?
Well, it's overwhelmingly through direct sexual transmission. That's the primary route, making it one of the most common sexually transmitted infections worldwide.
Can it be spread in other ways? Like sharing towels?
Indirect contamination, say via contaminated linen like damp towels or shared washcloths, or even things like toilet seats, has been described in the literature. However, this is considered much less common than sexual transmission, simply because the parasite survives so poorly outside the human body. The main focus for prevention and control has to be on sexual transmission.
Understood. What about the clinical presentation? Does it differ between men and women?
Yes, it does, and that's a really important distinction for diagnosis and for understanding its public health impact. In women, the parasite can be found in several locations, primarily the vagina, but also the urethra, sometimes the rectum, and even in glands near the vaginal opening, like the Skene's and Bartholin's glands.
And the symptoms in women?
Symptoms often involve what's called vulvovaginitis, inflammation of the vulva and vagina. This could be subacute or chronic. The hallmark symptom is often a persistent, abundant vaginal discharge, often called leukorrhea.
What's the discharge like?
It's frequently described as foul-smelling, and the color can vary, often whitish or yellowish, but sometimes it can be frothy or even greenish. This is usually accompanied by intense vulvovaginal itching or irritation, sometimes pelvic pain, and often dysuria, which is burning or pain during urination.
It sounds very uncomfortable.
Extremely. And during a clinical examination with a speculum, the healthcare provider often sees an intensely inflamed vaginal lining. Sometimes there are characteristic tiny red spots on the cervix, called strawberry cervix, or colpetus macularis, though this isn't always present. The inflammation can make the speculum insertion itself very painful for the patient.
Okay, that's in women. What about in men?
In men, the parasite can infect the urethra, the prostate gland, or the seminal vesicles. Now, here's a key difference. Infections in men are very often latent or asymptomatic, meaning they have the infection, they can transmit it, but they experience few or no obvious symptoms.
So men often don't know they have it.
Frequently, yes. If symptoms do occur in men, they typically present as urethritis, inflammation of the urethra. This might cause a discharge, which can be thin or purulent, and burning during urination.
Are there complications in men?
Yes, complications can occur, although they might be less common or less recognized than in women. These can include balanitis, inflammation of the head of the penis, cystitis, bladder infection, and prostatitis, inflammation of the prostate.
The high rate of asymptomatic infection, especially in men, must make control really difficult.
It poses a huge challenge for controlling its spread. Asymptomatic individuals, often men, act as unknowing reservoirs, continuing the chain of transmission. This is why partner notification and treatment are so critical for Trichomonas.
Right. So how is it diagnosed then, if symptoms can be absent or mimic other things?
Diagnosis relies on detecting the parasite, that vegetative trophozoite form, in genital samples. And because the parasite is so fragile, the conditions under which the sample is collected and handled are absolutely crucial for getting an accurate result.
What are those conditions?
Ideally, the sample should be taken before the patient starts any local treatment, like creams or douches, or any systemic antibiotics or endoparasitics. They should avoid any vaginal hygiene products right before the visit. And usually, 48 hours of sexual abstinence is recommended before sampling.
And getting it to the lab quickly.
Yes. Samples must be transported rapidly to the lab and examined promptly, or placed in special transport media if delay is unavoidable, to keep the trophozoites alive and mobile.
What kind of samples are taken?
For women, samples are typically collected during a pelvic exam using a speculum. Swabs are taken from the posterior vaginal fornix, the cul-de-sac, the vaginal walls, and sometimes the urethra or Bartholin's glands. For men, sampling often involves swabbing the urethra, usually early in the morning before urination, or examining the first void urine sediment. Sometimes examining secretions obtained after a prostatic massage is done.
And how is the sample examined in the lab?
The quickest and most common method is direct fresh microscopic examination, often called a wet mount or wet prep. A drop of the vaginal discharge or sediment is mixed with saline on a slide, covered with a cover slip, and examined immediately under a microscope.
What are they looking for?
They're looking for those characteristic rounded or pear-shaped mobile trophozoites, often with their jerky movement and visible flagella. Seeing the live motile organism provides a definitive diagnosis. Staining techniques like GIMSA or MGG can also be used on dried smears, which can help visualize the parasite structure but doesn't show motility.
Are there more sensitive tests if the wet mount is negative?
Yes. If clinical signs strongly suggest trichomoniasis, but the direct microscopic exams are negative, other methods are available. Culture on specialized media was considered a gold standard for a long time, as it can detect low numbers of parasites, but it takes several days to get a result. Nowadays, nucleic acid amplification tests, or NATs, which include PCR techniques, are becoming much more common. These tests detect the parasite's genetic material and are highly sensitive and specific. They can often be done on the same samples used for other STI testing, like urine or vaginal swabs. NATs are particularly useful when clinical suspicion is high, but parasites aren't seen on microscopy.
Okay, highly sensitive tests are available. What about the treatment?
For treatment, just like with Giardiasis, nitro-imidazole drugs are the mainstay. Metronidazole is again the most commonly proposed molecule.
Is the treatment course similar?
It's often given orally. A typical regimen might be, say, 250 milligrams 3 times a day for about 7 to 10 days. However, single-dose therapy is also very common and effective for trichomoniasis, often using a higher dose of metronidazole, like 2 grams, or tinidazole or secnidazole. Single-dose regimens can be really helpful for ensuring adherence, making sure the person completes the full treatment. Nemorazole is another option used in some places.
You mentioned partner treatment being mandatory.
Absolutely critical. Because it's an STI and asymptomatic infection is common, especially in male partners, it is mandatory to treat both partners simultaneously. Even if one partner tests negative or has no symptoms, they should be treated to prevent reinfection of the index patient and to stop further spread in the community. This is a core principle in managing STIs.
Should local treatment be added for women?
Yes. Alongside the oral medication, it's often recommended to add a local treatment for women, such as vaginal suppositories or ovules containing metronidazole or another anti-trichomonal agent. This can help resolve local symptoms more quickly and improve cure rates.
Makes sense. And prevention or prophylaxis?
Prophylaxis really boils down to adhering to the precautions that are common to preventing all sexually transmitted infections. The most effective measure is consistent and correct use of condoms during sexual intercourse. Reducing the number of sexual partners and mutual monogamy with an uninfected partner also reduce risk. Open communication with partners about sexual health and getting tested regularly are also key components. Basically, it underscores the importance of ongoing education about safe sex practices and public health efforts.
Right. Standard safe sex precautions are key. Okay, we've covered Giardia in the gut and Trichomonas in the urogenital tract. Now, you mentioned something a little different earlier, a ciliate.
Yes, exactly. Now for something a bit different. Let's shift our gaze slightly from the flagellates to their cousins, the ciliates. While they are generally less common causes of human disease than the flagellates we've discussed, there's one in particular, Balantidium coli, that provides a really fascinating glimpse into zoonotic infections.
Zoonotic meaning from animals.
Precisely. Balantidium coli causes a disease called Giardiasis. And what's particularly interesting here is that this parasite is primarily found in pigs. They are the main reservoir.
Pigs. So how do humans get it?
Humans typically get infected by accidentally ingesting the infective cyst stage. This usually happens through consuming contaminated food or water. Specifically, it's often linked to eating undercooked pork or perhaps charcuterie that has been contaminated. It can also happen if drinking water or other food items are soiled by pig feces containing the cysts.
So a direct link between pig hygiene, food handling, and human infection.
A very clear link. It really highlights that connection between animal health, food safety practices, and human well-being. It's a reminder that what happens in the pigsty can, quite literally, end up causing problems for us.
Okay. Does Balantidium coli also have different forms, like Giardia?
Yes, it does. Like many protozoa, it presents in two main forms. There's the vegetative form, which is the active feeding stage, and Balantidium coli trophozoites are quite large for a protozoan, often measuring between 50 to 100 micrometers, sometimes even larger. They're easily visible under the microscope.
And being a ciliate means?
Being a ciliate means its entire surface is covered in numerous short, hair-like structures called cilia. These bead in coordinated waves, allowing the parasite to move quite rapidly and purposefully, often in a boring or rotating motion as it navigates through the intestinal contents. It also has a distinct cytostome, or mouth, for ingesting food particles, like bacteria and debris.
Okay, that's the active form. What about the cyst?
Then there's the cystic form. This is the infective and resistant stage. It's generally more rounded or spherical than the trophozoite and slightly smaller, typically measuring between 50 to 60 micrometers in diameter.
Does it have cilia?
Usually the cyst stage is non-ciliated, or the cilia are retracted beneath a thick, protective cyst wall. This wall is what allows it to survive outside the host in the environment, waiting to be ingested, similar in function to the Giardia cyst.
Got it. So what happens clinically if someone gets infected with Balantidium coli?
The clinical picture for Balantidiasis can vary quite a bit. Just like with Giardia, some individuals can be asymptomatic carriers. They host the parasite, shed cysts in their feces, but don't show any signs of illness themselves. They can still spread it, though.
And if they do get symptoms?
When symptomatic, the infection typically manifests in the large intestine. It usually presents in one of two forms. The more common is the chronic form. This is often characterized by intermittent diarrhea, alternating with periods of constipation. The diarrheic episodes might occur in flares over time, accompanied by abdominal discomfort, nausea, and sometimes weight loss. It can be a persistent and uncomfortable condition.
Okay, chronic intermittent diarrhea. What's the other form?
The other form is the acute form, which is generally rarer but can be much more severe. Acute Balantiasis can present like dysentery, with frequent watery, sometimes bloody and mucoid stools, along with abdominal pain, cramping, fever, and dehydration.
Dysentery, like amoebic dysentery.
Exactly. It can actually mimic severe intestinal amoebiasis quite closely. This is because Balantidium coli, being large and motile, can invade the intestinal lining, causing ulcerations and inflammation, sometimes leading to flask-shaped ulcers similar to those seen with Entamoeba histolytica. It can even provoke numerous abscesses in the intestinal wall in severe cases. This makes accurate diagnosis really crucial to ensure the right treatment is given.
So diagnosis is important to differentiate it. How is it diagnosed?
Diagnosis is usually quite straightforward, thankfully. It's based on identifying the parasite in stool samples through standard parasitological examination.
Looking for the trophozoites or cysts?
Yes. Laboratory technicians look for either the large ciliated motile trophozoites or the characteristic rounded cysts in fresh or preserved stool specimens examined under the microscope. Because the parasite is so large, it's often relatively easy to spot compared to some of the smaller protozoa.
Okay, and treatment. What's used for Balantidiasis?
For treatment, the drug of choice is often an antibiotic from the tetracycline class, such as tetracycline itself or doxycycline. These are typically given for about 10 days.
Are there alternatives?
Yes, metronidazole, which we've discussed for Giardia and Trichomonas, is also effective against Balantidium coli and is often used as an alternative, especially if tetracyclines can't be used, like in young children or pregnant women. Sometimes, if the presentation is severe and mimics amoebiasis, treatment might involve a combination of drugs.
And prevention, given the link to pigs.
Prophylaxis for Balantidiasis really centers on preventing the ingestion of cysts from the pig reservoir. Key measures include ensuring pork is thoroughly cooked before consumption. Avoid undercooked or raw pork products. On a larger scale, rigorous veterinary control of pigs raised for meat is important, including good sanitation practices in pig farming to reduce contamination of the environment with feces. And as always, general collective and individual hygiene measures are vital things like protecting water supplies from fecal contamination and practicing good hand hygiene, especially for people who work closely with pigs.
Right, breaking that chain from pig feces to human ingestion. Fascinating. Okay, we've covered the big three: Giardia, Trichomonas, and Balantidium. What about other flagellates found in humans? Are there others we should know about?
Absolutely. While we've deep dived into the major players that cause significant disease, it's really important to remember there's a whole supporting cast, if you will, of other intestinal flagellates that can be found in humans. Most of these are generally considered to have little or no pathogenicity. They're often thought of as commensals, just living alongside us without causing harm.
So our internal world is quite a bustling ecosystem.
It really is.
Mhm.
And knowing about these other residents is useful, partly because they might show up in stool tests and need to be differentiated from the pathogens, and partly because, well, the status of some of them is still a bit debated.
Debated. Like which ones?
Well, a prime example is Dientamoeba fragilis. For a long time, it was considered non-pathogenic. However, its pathogenicity, its ability to cause disease, is actually quite controversial now and widely debated in the scientific community.
Why the controversy?
Because increasingly, many researchers and clinicians do consider it potentially pathogenic. It's been implicated in causing symptoms like persistent diarrhea, abdominal pain, bloating, and fatigue, particularly in children. Some studies have even linked it to the development or exacerbation of irritable bowel syndrome, IBS, much like another controversial gut protozoan, blastocystis.
Interesting. What's unusual about Dientamoeba?
What's truly interesting biologically about Dientamoeba fragilis is that despite its name suggesting it's an amoeba, it's actually classified as a flagellate, related to Trichomonas. However, it typically exists only in a vegetative amoeboid form in the gut. It usually lacks external flagella, although internal flagellar structures are present. And here's the really puzzling part. No definite resistant cystic form has ever been clearly described for Dientamoeba.
No cyst stage, like Trichomonas.
Exactly, which again raises the question of how it transmits effectively via the fecal-oral route if its trophozoite stage is presumed to be fragile.
So how is it thought to be transmitted?
That's still poorly understood, Malcolm knew, as the French sources might say. One fascinating and long-standing hypothesis suggests that it might actually hitch a ride inside the eggs of pinworms, Enterobius vermicularis.
Inside pinworm eggs? Seriously?
It's a leading theory. The idea is that the pinworm eggs, which are very resistant, could protect the fragile Dientamoeba trophozoites during passage through the environment and the stomach, allowing transmission. It's a remarkable example of potential interparasite cooperation, if true.
That is fascinating. Is it easy to diagnose?
Diagnosis can be challenging precisely because the trophozoite forms are so fragile and can disintegrate quickly in stool samples after they're passed. It often requires rapid examination of fresh stool or preservation in special fixatives immediately after collection. Permanent staining techniques are often needed to reliably identify its characteristic features, especially its nuclei. Often, too, hence the dye in Dientamoeba.
Okay, so Dientamoeba fragilis is one to watch with controversial pathogenicity. What are some of the other, more clearly non-pathogenic ones?
Sure, there are several others commonly encountered, generally considered harmless commensals. For example, there's Enteromonas hominis. This is a very small, non-pathogenic flagellate that lives in the cecum, part of the large intestine. It does form a cyst, but the cyst is tiny, only about 4 micrometers.
Okay, Enteromonas hominis. Any others?
Then there's Trichomonas intestinalis, which is now more commonly called Trichomonas hominis. This one was once considered perhaps the most frequent intestinal flagellate found in humans, especially prevalent in warmer, humid climates. Like its cousin Trichomonas vaginalis, it exists only as a vegetative trophozoid and never forms a cyst. Despite being common, it's generally regarded as non-pathogenic.
So another Trichomonas species, but in the intestine and harmless?
Generally considered so, yes. And just to add to the Trichomonas family diversity, there's also Trichomonas tenax, which is a flagellate found specifically in the human oral cavity in the mouth, around the teeth and gums. Also usually considered harmless, though sometimes associated with poor oral hygiene.
In the mouth. Okay, what else?
Another common one is Chelamastix misnili. This is considered a harmless saprophyte living off debris in the large intestine, particularly the cecum. Its vegetative form is noted for having a characteristic stiff, rotary, or corkscrew-like movement. And it forms a very distinctive lemon-shaped or pear-shaped cyst, which has a little anterior knob or protrusion. Interestingly, this cyst is often described as being the smallest of the protozoan cysts found in human feces.
Lemon-shaped cyst, corkscrew movement. Quite distinct.
Yes. And finally, there's Retortimonas intestinalis, another inhabitant of the large intestine, also generally considered non-pathogenic. It has both a vegetative trophozoite form and a small, pear-shaped cystic form.
So quite a diverse little community living in there, most of them not causing any trouble.
Exactly. These examples really demonstrate the incredible diversity of microscopic life within us. It reminds us that our gut microbiome isn't just bacteria, it includes these protozoa too. And importantly, not all tiny organisms we find are harmful. Many simply coexist with us as part of our normal internal ecosystem. Differentiating the pathogens from the harmless commensals is a key task in diagnostic parasitology.
What an absolutely eye-opening journey through this microscopic world, seriously. From the incredibly resilient cysts of Giardia, the ones that can survive chlorination and really challenge our assumptions about water safety,
Right, a major public health concern right there.
to the surprisingly fragile, sexually transmitted Trichomonas vaginalis that manages to spread so effectively despite having only that active form.
A totally different survival strategy relying on direct contact.
And then even the unique pork-borne Ciliate coli, linking animal husbandry directly to human illness. We've truly seen incredible diversity in how these tiny organisms live, how they reproduce, and sometimes how profoundly they can impact us, often completely unseen.
It really is remarkable. And, this raises an important question, doesn't it? How much more is actually out there thriving in these invisible ecosystems within us and around us that we don't fully understand yet?
That's a great point.
The kind of nuanced details we've discussed today, like the still controversial pathogenicity of Dentamoeba fragilis, or the very specific and unique survival strategies that each of these organisms has evolved, they really underscore the immense complexity of parasitology. And they highlight the ongoing importance of continued research and also of maintaining vigilant public health measures to protect our communities from these microscopic but sometimes powerful forces.
Absolutely. So thinking back over everything we've covered, what stands out most to you from this deep dive? What should our listeners really take away? Maybe think about the everyday implications of what we've discussed, the practical side of it.
Yeah, I think for me, it's the reminder of the hidden connections, how something seemingly simple like washing hands or cooking meat properly connects directly to preventing infection by Giardia or Balantidium, how water quality is constantly challenged by organisms like Giardia, and how personal health choices in the context of STIs directly impact the spread of Trichomonas. It's all interconnected.
Well said. Okay, for your final provocative thought to leave our listeners with, the next time you maybe consider the safety of your drinking water, especially if you're traveling or outdoors, or you think about the real importance of thorough hand hygiene, not just a quick rinse, or perhaps even ponder the mysterious origins of a persistent stomachache or unusual symptoms. Remember the tiny, powerful forces we've explored today. Our health is so intimately connected to these invisible worlds in ways we often don't even realize. It really reminds us that knowledge truly is power, even perhaps especially when dealing with life at the microscopic level. Understanding these organisms helps shape how we navigate our daily lives and how we can better safeguard our own well-being and the health of our communities.